So we're going to be talking about the DNA replication fork in prokaryotes using divorce as an anecdote. So DNA is very tightly packed and supercoiled. So in order for replication to happen, we need to unwind or release that supercoiling ahead of the replication fork, which you see here is done by gyrase. I like to call that our little trust issues in our little relationship here. Slowly unwinding it. And then in order for the replication to actually happen, we need helicase here, this little pink wedge, to break the hydrogen bonds between the nucleotides in DNA, which are adenine, thymine, guanine, and cytosine. I like to call that our little mistress here that breaks these bonds without, repair and keeps them kind of wedged apart and separated. Something else that helps kind of stabilize is protect our single strands here are our single-stranded binding proteins, which I like to think are our friends. They help prevent our single strand to become a double strand or hairpins for happening. The next thing that'll happen is our lawyer guy here, our DNA primase, will lay down RNA primers on both the leading strand and the lagging strand here, just so that DNA polymerase 3, our new love interest, can bind and start replicating. On our leading strand, it'll synthesize from 5' to 3' continuously towards this helicase into the replication fork. On our lagging strand that's not doing so hot because it'll synthesizing 5' to 3' going this way, it'll replicate in small fragments. That DNA polymerase 3 will make these small little fragments here called Okazaki fragments, which I like to think is, their life is in a bit of a shamble and some fragments there. The next thing that'll happen is our new job, our DNA polymerase 1, will go in and replace those RNA primers with DNA nucleotides. To finally seal up these little nicks or missing sugar phosphate bonds. We'll have the DNA ligase, which seals these little nicks and kind of fixes the rest of this DNA. I like to think of that as our kids here, kind of filling in the voids here. Something else about DNA is that it's composed of a sugar phosphate backbone that are connected by phosphodiester bonds. These phosphodiester bonds link up the 3' end of one nucleotide to the 5' end of the other and are usually cut for the origin of replication, or as I say, the breaking point in this marriage, this messy divorce. Other than that, I think that's about it.